I'm an associate professor of molecular biotechnology at the Turkish-German University. This masterclass is about computational approaches for aquaculture systems. There are many various computational approaches that can be categorized as big data analysis, computational biology, bioinformatics, classical machine learning, quantum computing, and as quantum machine learning. In this class, I will be relating these computational approaches to metagenomic studies in aquaculture systems. Metagenomics is the study of genetic material recovered directly from environmental samples, and it plays a crucial role in understanding microbial communities in various settings, including aquaculture systems. In aquaculture, metagenomics can be used to investigate diversity, function, and dynamics of microbial populations in water and sediment, as well as in the guts of farmed species. Specifically, metagenomics helps in identifying the wide range of microorganisms present in the aquaculture environment, which is essential for maintaining ecosystem health. Understanding microbial diversity can help in assessing water quality and detecting harmful pathogens. In fact, metagenomics can be used to monitor and detect pathogens that cause diseases in farm species. Furthermore, it helps in understanding the roles of microbes in nutrient cycles and their ability to break down waste products. Additionally, monitoring for antibiotic resistance genes can be helpful in assessing the impact of antibiotic use in aquaculture and guiding regulations to minimize the development of resistant bacterial strains. When we look at the relationship between metagenomics and aquaculture systems, metagenomics can be useful in microbiome analysis. Metagenomics enables to characterize the composition, diversity, and function of the microbial communities present in aquaculture systems by sequencing and analyzing the microbiome of water, sediment, and organisms in aquaculture environments. We gain insights into the interactions between microorganisms, host organisms, and the surrounding ecosystem. Metagenomic studies are important for disease diagnosis and disease management. Namely, metagenomic studies can contribute to the early detection and monitoring of microbial pathogens in aquaculture systems. By analyzing the metagenomics data from diseased organisms or environmental samples, we can identify potential pathogens, assess their virulence behavior, and develop targeted strategies for disease prevention and control in aquaculture operations. Furthermore, metagenomics plays a role in the discovery and development of probiotics and beneficial microorganisms that can improve the health and performance of aquaculture species. By analyzing the microbial communities in aquaculture systems, we can identify potential probiotic strains that promote growth, enhance disease resistance, and maintain water quality. Also, metagenomic studies can help in the selection of microbial species for bioremediation purposes, such as nutrient cycling and waste treatment in aquaculture systems. Finally, metagenomic studies provide valuable information for assessing the impact of aquaculture activities on the surrounding ecosystem. By evaluating the microbial diversity and functional potential of aquatic environments through metagenomics analysis, we can evaluate the environmental sustainability of aquaculture practices and develop strategies to mitigate potential risks to biodiversity and ecosystem health. So how can we help out in such studies, very easy. Metagenomic studies involve sequencing and analyzing the DNA from environmental samples, which can encompass a vast array of organisms from a particular habitat, such as an aquaculture site. This generates enormous volumes of data, fitting well into the domain of big data. So, what is big data analysis and how can it be helpful? The processing and analysis of metagenomic data requires sophisticated computational tools and algorithms, many of which are developed within the big data field. These tools can handle the sorting, mapping, and aligning of sequence data to reference genomes, identifying genetic material from a mixture of different species. Big data technologies offer solutions for storing and managing large data sets effectively. Databases and data lakes are used to maintain metagenomics data in a manner that is accessible and retrievable for analysis. Furthermore, big data analysis enables the discovery of patterns and sequences that represent biological significance in metagenomics data. Through machine learning, which are part of big data analytics, we can identify trends and gain insights about microbial communities, gene expression, and ecological interactions in aquaculture environments. By applying big data machine learning models to metagenomic data sets, we can predict how changes in the aquaculture environment, like temperature, salinity, and pollutants, might affect microbial communities and consequently the health and growth of aquaculture species. Big data analysis helps in optimizing feeding strategies, monitoring health conditions, and improving breeding techniques in aquaculture by analyzing the genetic and microbial components involved. So what is actually big data? Big data is a set of structured, semi-structured, and unstructured data produced in high volume, speed, and diversity. Big data is a new concept that describes heterogeneous data in different volumes that cannot be processed using traditional database techniques and consists of various digital contents. Structured data refers to all data types that are easy to model, input, store, query, process, and visualize. In general, they are presented in predefined fields of certain types and sizes and can be managed in relational databases or tables. In this data type, which has a solid structure, it is easier to obtain useful information compared to other data types, since the processes do not require high performance capabilities or parallel techniques. Semi-structural or self-describing data reflects a structured data type, but does not essentially contain a solid model. In other words, semi-structured data contains models in which structure is defined, as well as various meta-models such as labels and marks used to define specific elements and a hierarchical representation of different fields in the data. The most well-known examples of semi-structured data include XML and JSON programming languages, for example. Unstructured data are record types that are presented and stored outside a defined format. It generally consists of free format text such as books, articles, documents, emails, and media files such as images, audio, and video. The difficulty of presenting this type of data in a strict manner has led to the emergence of new mechanisms in data processing. There are three main components, the three V's, that characterize the big data phenomenon: variety, velocity, and volume. In addition to the three V that defines big data, In some sources, 6V is mentioned instead of 3V by including the veracity and value components. Data diversity refers to data that is produced in different types and formats and does not have a standard order. This diversity can be classified as structured, semi-structured, and unstructured data and may require different approaches to analyze and store and to store. Big data velocity refers to the speed at which data is generated. Today, data is often produced in real time or near real time, and therefore it must also be processed, accessed, and analyzed at the same rate to have any meaningful impact. Information is heterogeneous, meaning it can emerge out of various sources and can be organized, unstructured, or semi-organized. More customary organized information is currently enhanced by unstructured text, pictures, sound, video documents, or semi-organized designs like sensor information that cannot be coordinated in a decent information pattern. While the total amount of data produced in human history until 2003 was 5 exabytes, today the same amount of data is produced in just 2 days. The veracity shows how accurate or reliable the big data is. Data must be reliable enough to be used in business decisions. The high diversity of big data makes it difficult to ensure the quality and reliability of the analyzed data. Data quality is evaluated with reality, which is a very important dimension of big data, because reliable models can only be produced with high-quality data. Big data are information assets defined by high volume, velocity, and diversity, and require specific technology and analytical methods. It was possible for these data to create economic value for organizations and society by analyzing them and obtaining valuable insights. The meaning of the data collected is constantly changing, and this change can lead to inconsistency over time because data collection methods and targeted information change. Raw data is not useful for research or for any organization. Data processing is the method of collecting raw data and converting it into useful information. It is usually performed in a step-by-step process by a team of data scientists and data engineers within an organization. Raw data is collected, filtered, sorted, processed, analyzed, stored, and then presented in a readable format. Data processing is important for organizations to create better business strategies and increase competitive advantage. By converting data into readable formats such as graphs, tables, and documents, employees at all levels of the organization can understand and use the data. The data processing cycle comprises a progression of steps where crude raw data, which we call input, is taken care of into a framework to create significant experiences, which we call yield. Each step is taken in a particular request, yet the whole cycle is rehashed in a cyclic way. Generally, there are 6 main steps in the data processing cycle. These steps consist of collection, preparation, input, data processing, output, and storage. The type of raw data collected has a huge impact on the output produced. Hence, raw data should be gathered from defined and accurate sources so that the subsequent findings are valid and useful. The goal of the preparation step is to remove bad data, which means unnecessary, incomplete, or inaccurate data, and start collecting high-quality information so that it can be used in the best possible way for business intelligence. In the input step, the raw data is changed over into machine-readable form and taken care of into the processing unit. This can be in the form of data entry through a keyboard scanner or any other input source. Data processing is a step where the raw data is exposed to different information handling strategies utilizing AI and computerized reasoning calculations to create a beneficial output. This step might differ marginally from one interaction to another, relying upon the wellspring of data being processed and the expected utilization of the output. The data is finally transmitted and displayed to the user in a readable form like graphs, tables, vector files, audio, video, documents. This output can be stored and further processed in the next data processing cycle. The last step of the data processing cycle is capacity, where data and metadata are put away for additional utilization. This considers fast access and recovery of data at whatever point required, and furthermore permits it to be utilized as contribution to the data processing cycle straightforwardly. There are different types of data processing based on the source of data and the steps taken by the processing unit to generate an output. There is no one-size-fits-all method that can be used for processing raw data. Batch processing, real-time processing, online processing, multi-processing, and time sharing. In addition, methods of data processing have also been used, and there are single-user programming, multiple programming, real-time processing, online processing, time-sharing processing, and distributed processing systems. Multiple programming provides the facility to store and execute more than one program in the central processing unit, which is CPU, simultaneously. Further, the multiple programming technique increases the overall working efficiency of the respective computer. Benefits of data processing are increased productivity and profits, limited operational costs, and improved data storage, distribution, and reporting. Data management object refers to the technical object at each stage in the information system. Data management functions as an information security strategy and ensures the normal operation of the system in case of loss of initial data by restoring it to its original state through backup and recovery technology. All user data must be stored as well as other system-related data. The purpose of data collection and management is to ensure the recovery of data so that the risk to the system can be reduced and important system resources can be protected. The basic technologies for computer data storage and management are complex visualization-based layout algorithm. Graphical representation enhances users' data processing speed by visualizing data analysis and applications. In automatic metadata collection, thanks to the storage of different pharmaceutical equipment capabilities and metadata support methods, metadata is automatically collected and data collection efficiency is increased. Users can manage workflows through the visual interface by configuring job sheets, increasing the usability of data management. Computer data correlation processing is divided into two parts. The one part of this is Bresenham algorithm, and the second part is the rectifier linear unit, which we call ELU. So when we continue with big data analysis, we see that the data processing in big data analysis involves cleaning the raw sequencing data to remove low-quality reads and contaminants, assembling the high-quality reads into longer sequences called contents, find the use of bioinformatics software to annotate genes and identify taxonomic compositions and functional capabilities of the microbial communities. Data Analysis in big data analysis can be categorized as set statistical analysis, meaning applying statistical methods to compare different samples or conditions within our data set, or usage of machine learning algorithms to find complex patterns in the data that may correlate with environmental factors or fish health outcomes, or network analysis, which involves the construction of interaction networks to understand the relationships between different microbial species or genes. So, we mentioned already the use of bioinformatics with big data and metagenomics. How can bioinformatics help out for real? Bioinformatics techniques are used to process and interpret the massive amounts of DNA or RNA sequences obtained from environmental samples. Bioinformatics tools help in assembling, aligning, and annotating metagenomic data, enabling to identify and characterize the microbial diversity present in a given sample. By predicting gene functions, metabolic pathways, and biological processes associated with the microbial communities in a given environment, bioinformatics tools help to elucidate the functional capabilities of the microbial populations. This information is necessary for understanding the ecological roles of microorganisms, their interactions, and their impact on the environment. Furthermore, bioinformatics is instrumental in conducting comparative analysis of metagenomics datasets from different samples or environments. By comparing the genetic content of microbial communities, we can identify conserved genes, unique features, and differences in functional potential across samples. These comparative genomic analysis provides insights into the adaptability, diversity, and evolutionary relationships of microbial populations in different habitats. Finally, bioinformatics resources, such as databases, software tools, and repositories, are invaluable for storing, managing, and analyzing metagenomics data. Specialized databases such as MGRS, IMGM, and Metabase provide access to curated metagenomics datasets, reference genomes, and analysis pipelines that enable us to explore and understand microbial communities. The advent of high-performance sequencing platforms has made it possible today for a complete genome to be sequenced by genomics laboratories in just a few days. The development of these high-performance sequencing platforms has necessitated the development of big data applications in the biological sciences. Large-scale experiments in medicine and biological sciences and new analysis methods have also made a significant contribution to big data. The data produced brings with it a number of problems. It has necessitated the necessity of a number of new technological developments, such as data processing, visualization, and storage. In terms of data analysis, developments in the field of biological sciences have also contributed to the development of modern bioinformatics. Technological and scientific developments have made it almost obligatory for all branches of science to work interdisciplinary. As a result of the increased integration of computer science with biology, new scientific fields such as computational biology and bioinformatics have emerged. The increase in studies, the development of analysis methods and experimental techniques have led to the formation of data in many different fields of biology. Analysis methods such as genomics, proteomics, molecular structure analysis, and the increase in the interactions of data sets obtained in these fields make it impossible to analyze data manually. The technologies developed in the high data processing step have led to the establishment of various databases for the storage of these data. These databases not only store the data, but also allow different researchers to utilize these data. National Center for Biotechnology Information, NCBI, European Bioinformatics Institute, EBI, and DNA Data Bank of Japan, DDBJ, are the most widely used databases today. There are also databases specialized in different fields. For example, the Encyclopedia of DNA Elements, ENCODE, and RefSeq for the human genome, and Ensemble database for comparative genomics and sequence variation studies on vertebrate genomes are frequently used. The stages of data generation start with experimental studies. Methods such as microarray and next-generation sequencing also generate large amounts of data. In addition, population surveys, patient registries, and clinical trials also generate big data. After the processes of collecting, obtaining, and processing data, these data need to be analyzed correctly. There are various tools for analyzing big data in bioinformatics. FastQC and Chemomatic tools are used for data control and filtering. Normalization, feature selection, and dimensional reduction operations should be performed on this data to bring the data obtained as a result of data control and filtering into a suitable format for further analysis. Commonly used tools for processing RNA sequence data are DESEC2 and HR tools. BIOAQUA, HiSET2, and STAR tools are commonly used for the alignment step, which is a critical step in the analysis of high-throughput sequencing data. While Lemma and HR tools are commonly used for statistical analysis of the resulting data. Big data analysis can be divided into two groups, actually, descriptive and predictive analysis. Descriptive analysis is the type of analysis that describes big data and enables the analysis of historical data, anomalies, similarities, or relationships between organisms. DNA, RNA, and protein sequence, gene expression data, protein-protein interaction data, gene ontology data, and pathway data are the 5 types of big data in bioinformatics. The biggest contribution to big data production in biological sciences is provided by sequence data, it can be said. Sequence data can provide information about the genome relationship or evolutionary links between organisms. Next-generation sequencing generates large amounts of data as it provides sequence information about the whole genome. Different tools are used to analyze sequence data. BLAST and Clustal Omega are used for alignment of sequence data. Galaxy and Strand NGS for NGS analysis, MapSplice and MEV for RNA-seq analysis. Understanding the progression of diseases or the effect of treatments applied against diseases can be learned through gene expression analysis by examining under what conditions genes are activated and under what conditions they are inhibited. For microarray-based gene expression profiling, there is a standard approach for recording gene expression levels. Anomalies in the interactions between proteins underlie many diseases. Due to the importance of these interactions, protein-protein interactions have been studied in many different fields, such as with molecular dynamics, bioinformatics, biochemistry, and quantum chemistry, resulting in high volume of heterogeneous data. And String 11 and BioGrid 12 are important repositories for protein-protein interactions. The Go database provides dynamic, structured, and time-independent gene ontologies for relevant biological processes, cellular components, and molecular functions. The database uses a controlled vocabulary to facilitate querying at all levels. There are also some tools for conducting bioinformatics studies using the Go database, such as Amigo, DAG-Edit, or OvoEdit, most of which are third-party based, but which the Go project itself also manages. Pathway analysis are of great importance for understanding the molecular basis of diseases, identifying genes and proteins related to the etiology of diseases, and predicting drug targets. KEC, reactome, and pathway commons are important pathway data sources. So, machine learning algorithms can be trained to classify and assign taxonomic labels to metagenomic sequence reads based on features extracted from the data. This can help in accurately identifying and characterizing the microbial taxa present in aquaculture environments. Furthermore, machine learning models can be used to predict the functional potential of microbial communities based on metagenomic data. By training algorithms on non-functional annotations, we can infer the metabolic capabilities and biological functions of microbial populations and aquaculture systems. In addition, machine learning techniques such as feature selection and classification algorithms can be employed to identify biomarkers associated with specific traits or conditions. This can help in biomarker discovery for monitoring the health, productivity, and sustainability of aquaculture systems. Moreover, machine learning algorithms can be used to develop predictive models for assessing the impact of microbial communities on aquaculture performance, disease outbreaks, or environmental sustainability. Finally, machine learning approaches can help in integrating and analyzing multi-omics data such as metagenomic, metatranscriptomic, and metabolomic data. The most widely used machine learning models in my group are random forest, which is a versatile and powerful assembled learning algorithm that can be used for classification, regression, and feature selection tasks in aquaculture metagenomic studies. It is known for its robustness, scalability, and ability to handle high-dimensional data. Moreover, we use support vector machines, which is a popular supervised learning algorithm that is commonly used for classification tasks in aquaculture metagenomic studies. Support vector machines is effective for dealing with complex data sets and can identify patterns and relationships within the data. Next, we have neural networks, which are deep learning models such as convolutional neural networks, recurrent neural networks. These models are capable of learning complex patterns and features from large-scale genomic data sets. A new technology that is used more and more currently is quantum computing. We started also with quantum computing studies. Quantum computing utilizes the principles of quantum mechanics to perform operations on data. Unlike classical computers, it uses qubits, which can exist in multiple states simultaneously thanks to superposition. It also takes advantage of entanglement, where qubits can be intricately linked to each other, allowing even for more powerful capabilities. Quantum computing has the potential to revolutionize aquaculture system studies. Quantum information processing is a new type of information processing based on quantum physics, which is in some ways more useful than classical information processing. It mainly uses quantum bits, which we call qubits, instead of bits in classical computers. While bits can only be two different values, 0 and 1, qubits can be a superposition of all computable states. Thus, a system with n qubits can exist in 2 to the n different superposition states. This means that a system with 2 qubits can take 4 different states. The major advantages of quantum information processing come from quantum parallelism, quantum interaction, and quantum entanglement, apart from the superposition that we stated before. Of these, quantum parallelism in particular allows for simultaneous computation, which is very important in big data and because processing large volumes of data in sequence would require a lot of time, and this is one of the major problems of big data processing on classical computers. Using classical machine learning methods poses difficulties in the face of ever-increasing data. Increasingly growing data is heading towards a point where it cannot be measured and analyzed for many classical machine learning methods. In order to cope with this problem, machine learning methods that provide exponential acceleration against classical algorithms have begun to be developed. There are two main approaches to developing quantum machine learning methods. The first of these approaches is to adapt classical machine learning methods to quantum machine learning. Algorithmic accelerations are aimed at this adaptation. Starting with this approach, quantum variants of many machine learning models have been created. For example, quantum support vector machine, quantum k-nearest neighbor, quantum neural network are the counterparts of classical machine learning methods in the quantum field. Another approach in the field of quantum machine learning is algorithms based on quantum subroutines. Grover's algorithm, variational quantum circuit, and HHL algorithm can be given as examples of these algorithms. Quantum pattern matching can enhance the efficiency of genetic assembly methods. Although quantum methods like Grover's search could theoretically replace traditional database lookup and crack, and practical implementation faced challenges due to state preparation and collapse issues. An alternative approach involves extending exact search and cracking to allow for fuzzy searching. Quantum associative memories can be employed for fuzzy retrieval of database entries, improving binning accuracy and resilience to sequencing errors. Special considerations for efficiency are crucial due to the expensive procedure of constructing the database state. It's proposed to utilize quantum search only when exact matching fails, thereby limiting queries to cases requiring fuzzy matching. Suggestions for adapting quantum methods for storing and retrieving DNA sequences have been outlined by us recently with a focus on appropriate metrics for efficiency testing. And when it comes to quantum machine learning combines the principles of quantum computing and machine learning, as we said. It uses superposition and entanglement to enhance the performance of machine learning algorithms. One main goal of mine is to develop algorithms that can take advantage of quantum parallelism and entanglement to solve optimization, pattern recognition, and classification more efficiently. So, when it comes to quantum computing and bioinformatics, quantum computing is related to bioinformatics and therewith to metagenomic studies. Quantum computing offers the potential to exponentially increase computational power compared to classical computers. Bioinformatics, which is the field that deals with the analysis of large biological data sets and analyze vast amount of genomic, proteomic, and other biological data much faster and efficiently, can benefit from quantum computing's computational power. Quantum computing is driving the new development of new quantum algorithms that can outperform classical algorithms in solving specific bioinformatics problems. We are working on how quantum computers and algorithms can be applied on tasks such as sequence alignment, protein structure prediction, molecular dynamics simulations, and drug discovery. Furthermore, bioinformatics relies heavily on sophisticated data analysis techniques to interpret biological information and extract meaningful insights. Quantum computing enhances data analysis by enabling the processing of complex data sets in parallel, allowing for more comprehensive analysis of genomic sequences, biomolecular interactions, and biological networks. Moreover, bioinformatics often involves solving optimization problems, such as genome assembly, protein folding, and network analysis. Quantum algorithms are more efficient than classical computers in optimization problems, and by leveraging quantum computing for optimization tasks, we can streamline data analysis workflows and accelerate research outcomes. When we use quantum machine learning in aquaculture system studies, this is an emerging and innovative approach that has the potential to revolutionize the study of microbial communities in aquatic environments. It can be helpful in optimization, disease detection, environmental monitoring, and resource management. So, how does quantum machine learning function for metagenomics analysis? Data collection is an important step. Collecting samples from the aquaculture system, such as water, sediment, or organisms, to obtain genetic material for metagenomic sequences is the first step. This provides us a snapshot of the microbial diversity and composition in the aquaculture environment. Next step involves metagenomic sequencing. Performing metagenomic sequencing to generate raw DNA sequences from the collected samples is crucial. Utilizing next-generation sequencing technologies to obtain high-throughput data that captures the genetic information of the microbial communities present in the aquaculture system. Then data pre-processing is a crucial step. Processing and cleaning the metagenomic data to remove sequencing errors, filter out low-quality reads, and trim adapter sequences are crucial. This step is essential for ensuring the accuracy and reliability of the subsequent analysis. Then we apply quantum machine learning algorithms for metagenomic analysis. Quantum machine learning techniques such as quantum neural networks, quantum support vector machines, and quantum clustering algorithms can be used to classify microbes, predict functional attributes of the microbiome, and uncover patterns in the metagenomic data. We can also extract relevant features from the metagenomic data using quantum machine learning methods. These features may include taxonomic information, such as species abundance, functional gene annotations, or microbial interactions within the ecosystem. Next, we train quantum machine learning models on the extracted features to predict microbial functions, classify microbial taxa, or identify biomarkers associated with specific traits or conditions in the aquaculture system. We evaluate the performance of the models using metrics such as accuracy, precision, recall, and F1 score. We can interpret the results obtained from the quantum machine learning analysis and visualize the relationships between different microbial taxa, functional pathways, and environmental variables in the aquaculture system. However, validating the findings from the quantum machine learning analysis using traditional bioinformatics tools, experiments, and field studies is very important. Data pre-processing in quantum machine learning is a crucial step, and I would like to provide some insights into it. First, quality control is important. For this purpose, check the quality of raw sequencing data by examining quality scores, base composition, and read length distribution using tools such as FastQC and MultiQC. Trim or filter out low-quality reads, adapter sequences, and ambiguous bases to improve the overall quality of the data. And then second, trim sequencing reads to remove adapter sequences, low-quality bases, and sequencing errors. Tools like Trimomatic and CutAdapt can be used to perform read trimming based on quality scores, read lengths, and sequence composition. Next, we have to align the clean reads to a reference genome or a database of non-microbial sequences using alignment tools like BIOAQUA, BWA, or HiSet2. This step helps in mapping the sequencing reads to non-microbial species and identifying potential contaminants. Next step is taxonomic classification. We have to classify the sequencing reads into taxonomic groups based on their similarity to reference databases such as Silva, Green Genes, and NCBI. Tools like CRAC and Metaphylan can be used for taxonomic classification of metagenomic data. Then functional annotation is an important step. We have to annotate the functional pathways and gene functions associated with the metagenomics data using tools like MGRAST, KEGG, or InterProScan. This step helps in understanding the metabolic potential and functional diversity of the microbial communities in the aquaculture system. We also identify and remove any potential contaminants or artifacts from the sequencing data. This can include host DNA, sequencing artifacts, and environmental contaminants. We normalize the metagenomic data to account for variations in sequencing depth, read length, and sampling biases. Normalization helps in comparing the abundance of microbial taxa and functional genes across samples accurately. We format the pre-processed data into a suitable input format for quantum machine learning algorithms. For this step, we have to convert the taxonomic or functional annotations into numerical values or features that can be used for model training and analysis. We split the pre-processed data into training, validation, and testing sets for model development and evaluation, and we do this randomly. So, quantum machine learning analysis for metagenomics data in aquaculture systems include data representation, meaning convert the pre-processed data into a suitable format for quantum machine learning analysis. This may involve encoding taxonomic information, functional annotations, or other microbial features into quantum computational states, which is qubits, to represent the biological data effectively. This step involves feature encoding and quantum state encoding. Then we go into quantum circuit design. We designed quantum circuits that can process and manipulate the encoded metagenomic data. For this purpose, we defined a quantum register that represents the qubits used to encode the biological features. We determined the number of qubits needed based on the complexity of the metagenomic data into specific analysis tasks. We mapped the pre-processed metagenomic data, such as taxonomic information or functional annotations, onto the qubits in the quantum register. We assigned quantum states to represent different biological features, such as microbial taxa, gene functions, or other properties. We select the quantum gates that perform the desired transformation on the qubits to manipulate and analyze the encoded biological data. We designed the quantum circuit to apply the selected quantum gates in a sequential order that transforms the encoded biological features and prepares the data for analysis. We determined the sequence of quantum gates that maximizes the performance of the quantum machine learning algorithm for the metagenomic analysis. Then we implement the quantum circuit using a quantum programming language or framework such as Qiskit or PennyLane. We write the code that defines the quantum operations, initializes the quantum register with the encoded data, and executes the circuit to analyze the metagenomic data. Next, we test and validate. We evaluate the performance of the circuit in processing the metagenomic data and generating meaningful insights about the microbial communities in the aquaculture system. We fine-tune the quantum circuit by adjusting the parameters, gate sequences, and data encoding schemes to optimize the performance and accuracy. We usually iterate on the circuit design based on feedback from the simulation results and experimental validation. Once the quantum circuit design is optimized and validated, we run the analysis on a quantum processor. We adapt the circuit for the specific quantum hardware architecture and constraints to perform real-world metagenomics analysis using quantum computing resources. So when it comes to quantum gates, I will list here 6 quantum gates. The first one is the Hadamard gate. The Hadamard gate is commonly used in quantum machine learning algorithms for creating superposition states. It prepares the qubits to explore multiple states simultaneously, which can be useful for parallel processing and exploring complex interactions between microbial features and metagenomic data. Then we have the Pauli X or NUT gate. The Pauli X gate is equivalent to the classical NUT gate and flips the state of the qubit between 0 and 1. It can be used for data manipulation, flipping the encoding of biological features to facilitate specific computational operations in the metagenomic analysis. The controlled NUT gate acts on 2 qubits, where one serves as the control qubit and the other as the target qubit. It flips the target qubit if the control qubit is in the state 1. CNUT gates are essential for entangling qubits and creating correlations between different biological features in the metagenomic data. Then we have the phase gate. Phase gates introduce phase shifts to qubits and can be used to adjust the relative phase of quantum states during the computation. By applying phase gates, we can manipulate the interference patterns of encoded biological features and enhance the performance of quantum algorithms for metagenomic analysis. Universal gates, such as Toffoli or Fredkin gates, enable the implementation of arbitrary quantum operations on multiple qubits and support more complex transformations in the quantum circuit. These gates can be used for advanced data manipulations and computations in metagenomics analysis. And finally, we have quantum variational circuits composed of parametrized gates and optimization layers. These offer flexibility in designing quantum algorithms for machine learning tasks. By choosing suitable variational forms and parameterized gates, we can adapt the circuit architecture to the specific requirements of the metagenomic analysis. So when it comes to the quantum machine learning algorithms that can be used, quantum support vector machine is one of them. This is a quantum-inspired version of the classical support vector machine algorithm that utilizes quantum computing principles to enhance classification tasks. It aims to separate data into different classes by maximizing the margin between them, leveraging quantum algorithms for efficient feature mapping and classification. It can be effective for classifying microbial taxa, predicting functional attributes of the microbiome, or identifying biomarkers associated with specific traits or conditions in the aquaculture system. It can leverage quantum computing principles to achieve efficient classification and optimization tasks, making them suitable for analyzing complex metagenomic data sets. Then we have quantum neural networks. These integrate quantum circuits within neural network architectures for enhanced learning and inference capabilities. It leveraged quantum properties to perform parallel processing, gradient descent optimization, and representation learning, enabling neural networks to benefit from quantum speedups and processing complex data structures efficiently. Quantum neural networks can offer enhanced learning capabilities and processing power for analyzing intricate microbial community structures, functional gene annotations, or interaction networks in aquaculture systems. These can handle complex data representations, facilitate feature extraction, and optimize neural network architectures for metagenomic analysis tasks. Then we have quantum variational algorithms, such as the variational quantum eigensolver and quantum approximate optimization algorithm. These apply variational principles in quantum computing to solve optimization problems and find the optimal configurations for a given objective function. Variational algorithms use parameterized quantum circuits to iteratively optimize solutions and achieve near optimal outcomes for complex optimization tasks. Then we have quantum Boltzmann machines. Quantum Boltzmann machines are quantum-enhanced versions of classical Boltzmann machines, which model complex probabilistic relationships in data. Quantum Boltzmann machines leverage quantum principles to explore high-dimensional probability spaces efficiently, enabling the generation of probability distributions and sampling from the learned representations for machine learning and data analysis applications. Quantum clustering algorithms, such as quantum k-means and quantum hierarchical clustering, leverage quantum computing principles to group data points into clusters based on similarity metrics. Quantum clustering algorithms optimize the arrangement of qubits to represent data points and measure similarities, enabling efficient clustering of high-dimensional data sets with quantum speedups. Quantum clustering algorithms such as quantum k-means or quantum hierarchical clustering can help identifying microbial clusters, community structures, or ecological niches in the aquaculture microbiome. These algorithms can efficiently group microbial taxa based on similarity metrics, reveal patterns in the metagenomic data that reflect the underlying microbial communities and their interactions. And then finally, we have quantum principal component analysis. These aim to reduce the dimensionality of data by projecting it onto a lower dimensional subspace while preserving the most relevant information. Quantum PCA techniques employ quantum algorithms to find the optimal principal components of the data, enabling efficient feature extraction and dimensionality reduction for machine learning tasks. Quantum principal component analysis techniques can assist in reducing the dimensionality of metagenomic data, extracting relevant features, and uncovering the key factors driving microbial diversity and community dynamics in aquaculture systems. Quantum principal component analysis can facilitate the visualization of high-dimensional data, identify important biological patterns, and enhance the interpretation of metagenomic analysis results. So in summary, in this lesson, we learned computational approaches like big data analysis, bioinformatics, a little bit computational biology, classical machine learning, quantum computing, and quantum machine learning. I would like to thank my co-worker, Semih Alpsoy, for helping me with this masterclass. And thank you very much for listening. I wish you a wonderful day or evening.